used amino acids which are needed to make proteins with passing sparks through a mixture of hydrogen, methane, ammonia, and water. Mixture of gases simulating atmosphere of early Earth. So, water vapor, spark simulating lightning storms, condensation chamber, cold water cooled chamber causing droplets to form, liquid containing amino acids and other organic compounds. So, a mixture of gases simulating atmosphere of early Earth means hydrogen, methane, ammonia, and water. For several reasons, atoms do not assemble themselves into complex organic molecules or living cells on Earth today. For one thing, the oxygen in the atmosphere is very reactive and would destroy any organic molecules that formed. In addition, as soon as organic molecules appeared, something, bacteria or some other life form, would probably eat them. But the early Earth was a very different place. Could organic molecules have evolved under those conditions? In the 1950s, American chemists Stanley Miller and Harold Urey tried to answer that question by simulating conditions on the early Earth in a laboratory setting. They filled a flask with hydrogen, methane, ammonia, and water to represent the atmosphere. They made certain that no microorganisms could contaminate the results. 그래서 일단 무슨 질문이었냐면 could organic molecules have evolved under those conditions? Which were those conditions? The oxygen in the atmosphere is very reactive, and as soon as molecules appeared, the other life form would probably eat them, bacteria-like things. They filled a flask with hydrogen, methane, ammonia, and water to represent the atmosphere. They made certain that no microorganisms could contaminate the results. Then, as shown in figure 17.8, they passed electric sparks through the mixture to simulate lightning. 번개. The results were spectacular. Over a few days, several amino acids, the building blocks of proteins, began to accumulate. Miller and Urey's experiments suggested how mixtures of the organic compounds necessary for life could have arisen from simpler compounds present on a primitive Earth. Scientists now know that Miller and Urey's original simulations of Earth's early atmosphere were not accurate. However, similar experiments based on more current knowledge of Earth's early atmosphere have also produced organic compounds. In fact, one of Miller's experiments in 1995 produced cytosine and uracil, two of the bases found in RNA. One of Miller's experiments in 1995 produced cytosine and uracil. 오, 그래요? 95년도 밀러의 실험에서는 사이토신과 유라실을 합성했어. How did life begin? A step of organic molecules is a long way from a living cell, and the leap from non-life to life is the greatest gap in scientific theories of Earth's early history. Scientists do know that about 200 to 300 million years after Earth cooled enough to carry liquid water, cells similar to modern bacteria were common. How might these cells have originated? 지구가 냉각되고 200에서 300만 년이 충분히 지났고 나서는 박테리아 같은 게 꽤나 흔해졌다. Formation of microspheres. Under certain conditions, large organic molecules can form tiny bubbles called proteinoid microspheres, as shown in figure 17.9. Proteinoid microspheres. Large organic molecules can sometimes form tiny protein microspheres like the ones shown here. Comparing and contrasting. How are proteinoid microspheres similar to cells? How are they different? 중요한 질문이네. 이렇게 called proteinoid microsphere. Proteinoid microsphere. Microspheres are not cells, but they have some characteristics of living systems. Like cells, they have selectively permeable membranes. 선택적 투과성막을 갖고 있고, through which water molecules can pass. Microspheres also have a simple means of storing and releasing energy. 간단한 에너지 방출 및 저장 방법을 갖고 있어. Several hypotheses suggest that structures similar to proteinoid microspheres might have acquired more and more characteristics of living cells. 일단은 첫 번째 선택적 투과성막을 갖고 있어. They have selectively permeable membrane and simple means of storing and releasing energy through membranes or something. Evolution of RNA and DNA. Another unanswered question in the evolution of cells in the origin of DNA and RNA. Remember that all cells are controlled by information stored in DNA, which is transcribed into RNA and then translated into proteins. How could this complex biochemical machinery have evolved? DNA는 저장, store, and RNA 전사가 transcribe and 번역이 translate into proteins. The information in DNA is transcribed into RNA and then it is translated into proteins. Science cannot yet solve this puzzle, although molecular biologists have made surprising discoveries in this area. Under the right conditions, some RNA sequences can help DNA replicate. Other RNA sequences process messenger RNA after transcription. 무슨 소리야? Under the right conditions, some 몇 개 RNA sequence는 DNA 복제를 돕고, other RNA sequences process messenger RNA after other RNA sequences process messenger RNA after transcription. process가 동사야? 천천히 걷다 이동하다. Process messenger RNA after transcription. Still others catalyze chemical reactions. Some RNA molecules can even grow and duplicate themselves, suggesting that RNA might have existed before DNA. RNA가 DNA의 전신일 수 있다 이거지. A series of experiments that simulated conditions of the early Earth have suggested that small sequences of RNA could have formed and replicated on their own. From this relatively simple RNA-based form of life, several steps could have led to the system of DNA-directed protein synthesis that exists now. This hypothesis is shown in Figure 1710. Future experiments are aimed at refining and retesting this hypothesis. So, this hypothesis is described here. A body stew of inorganic matter, simple organic molecules, RNA nucleotides, RNA able to replicate itself, synthesize proteins, and function in informative storage. And by some means, proteins build cell structures and catalyze chemical reactions. RNA helps in protein synthesis. DNA functions in informative storage and retrieval. One hypothesis about the origin of life illustrated here suggests that RNA could have evolved before DNA. Scientists have not yet demonstrated the later stages of this process in a laboratory setting. How might RNA have stored genetic information? 이거는 염기서열 가지고 했다라고 설명하면 되거나. Proteins build cell structure and catalyze chemical reactions. Free oxygen. Ancient photosynthetic organisms produced a rise in oxygen in Earth's atmosphere. These rock-like formations called stromatolites were made by cyanobacteria, which were among the earliest organisms to evolve on Earth. The stromatolites above are growing in the ocean near Australia. Free oxygen, microscopic fossils or microfossils of single-celled prokaryotic organisms. That resemble modern bacteria have been found in rocks more than 3.5 billion years old, as shown in Figure 1711. Those first life forms must have evolved in the absence of oxygen because Earth's first atmosphere contained little or none of that highly reactive gas. 일단은 prokaryotic. prokaryotic 원생생물인가? 원핵생물. 블랙베리는 또 소리가 안 나. Over time, as indicated by fossil evidence, photosynthetic bacteria became common in the shallow seas of the Precambrian. By 2.2 billion years ago at the latest, these organisms were steadily churning out oxygen, an end product of photosynthesis. One of the first things oxygen did was to combine with iron in the oceans. 중요해요. 바닷속에 철과 결합했어. In other words, it caused the oceans to rust. When iron oxide was formed, it fell from the sea water to the ocean floor. There it formed great bands of iron that are the source of most of the iron ore mined today. 호상철광층. Without iron, the oceans changed color from brown to blue-green. Next, oxygen gas started accumulating in the atmosphere. As atmospheric oxygen concentrations rose, concentrations of methane and hydrogen sulfide began to decrease. The ozone layer began to form, and the skies turned their present shade of blue. Over the course of several 100 million years, oxygen concentrations rose until they reached today's levels. Next, oxygen gas started accumulating in the atmosphere. 그래서 상대적으로 메테인이랑 and hydrogen sulfide면 H2SO4인가? CH4 and H2SO4 이산화황이겠지? 하이드로젠 설파이드가 그럼 뭐지? 하이드로젠 설파이드 황화수소 H2S The increase in this highly reactive gas created the first global pollution crisis. To the first cells, oxygen was a deadly poison. The rise of oxygen in the atmosphere drove some life forms to extinction, while other life forms evolved new, more efficient metabolic pathways that used oxygen for respiration. Organisms that had evolved in an oxygen-free atmosphere were forced into a few airless habitats where their anaerobic descendants remain today. Some organisms, however, evolved ways of using oxygen for respiration and protecting themselves from oxygen's powerful reactive abilities. This stage was set for the evolution of modern life. What process added oxygen to Earth's atmosphere? 이것은 광합성을 하는 cyanobacteria. The photosynthesis of cyanobacteria in the early Earth. Origin of eukaryotic cells, 진핵세포. Several important events in the history of life have been revealed through molecular studies of cells and their organelles. One of these events is the origin of eukaryotic cells, which are cells that have nuclei. Eukaryotic cells are the cells that have nuclei. About 2 billion years ago, prokaryotic cells without nuclei, began evolving internal cell membranes. The result was the ancestor of all eukaryotic cells. Then something radical seems to have happened. Other prokaryotic organisms entered these ancestral eukaryote. These organisms did not infect their host as parasites would have done, and the host did not digest them as it would have digested prey. Well, instead, the smaller prokaryotes began living inside the larger cell, as shown in figure 1712. Over time, a symbiotic or interdependent relationship evolved. According to the endosymbiotic theory, eukaryotic cells formed from a symbiosis among several different prokaryotic organisms. One group of prokaryotes had the ability to use oxygen to generate energy-rich molecules of ATP. These evolved into the mitochondria that are now in the cells of all multicellular organisms. Other prokaryotes that carried out photosynthesis evolved into the chloroplasts of plants and algae. The endosymbiotic theory proposes that eukaryotic cells arose from living communities formed by prokaryotic organisms. This hypothesis was proposed more than a century ago when microscopists saw that the membranes of mitochondria and chloroplasts resembled the plasma membranes of free-living prokaryotes. Yet, the endosymbiotic theory did not receive much support until the 1960s, when it was championed by Lynn Margulis of Boston University. She and her supporters built their argument on several pieces of evidence. First, mitochondria and chloroplasts contain DNA similar to bacterial DNA. Second, mitochondria and chloroplasts have ribosomes whose size and structure closely resemble those of bacteria. Third, like bacteria, mitochondria and chloroplasts reproduce by binary fission when the cells containing them divide by mitosis. Thus, 세 가지 근거를 들었군, 보스턴 대학에서. The endosymbiotic theory. This endosymbiotic theory. 근거로 첫 번째는 DNA 구조가 유사하다. 미토콘드리아랑 엽록체의 멤브레인에 아니지, DNA 박테리아 유사. 두 번째, 미토콘드리아랑 엽록체는 리보솜의 구조가 비슷하다. 엽록체가 리보솜 크기와 구조가 박테리아와 유사. 3번. Third, like bacteria, mitochondria and chloroplasts reproduce by binary fission. 이분법으로 분열한다. 미토랑 엽록체가 세포 분열 시 이분법으로 분열한다. Binary fission. Reproduce by binary fission when the cells containing them divide by mitosis. These mitochondria and chloroplasts have many of the features of free-living bacteria. These similarities provide strong evidence of a common ancestry between free-living bacteria and the organelles of living eukaryotic cells, sexual reproduction, and multicellularity. Sometime after eukaryotic cells arose, those cells began to reproduce sexually. This development enabled evolution to take place at far greater speeds than ever before. How did sexual reproduction speed up the evolutionary process? Most prokaryotes produce asexually. Often they simply duplicate their genetic material and divide into two new cells. Although this process is efficient, it yields daughter cells that are exact duplicates of the parent cell. Thus, this type of reproduction restricts genetic variation to mutations in DNA. 그래서 이런 무성 생식은 DNA의 변이나 변화가 적고, sexual reproduction, on the other hand, shuffles and reshuffles genes in each generation, much like a person shuffling a deck of cards. The offspring of sexually reproducing organisms, therefore, never resemble their parents exactly. This increase in genetic variation greatly increases the chances of evolutionary change in a species due to natural selection. A few 100 million years after the evolution of sexual reproduction, evolving life forms crossed another great threshold. the development of multicellular organisms from single-celled organisms. In the blink of an evolutionary eye, these first multicellular organisms, such as one shown in Figure 1713, experienced a great increase in diversity. The evolution of life was well on its way. The ancient jellyfish, 원색 해파리, 고대 해파리는 an early multicellular animal from Precambrian time, did not have bones or other hard parts, but it left behind a fossil that allowed biologists to infer its overall shape. What evidence shows that this organism had body parts arranged around a central point?